With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Question here says, after insertion in activation of galactosidase gene, which type of colonies do not produce any color? First of all, what is this insertion in activation of galactosidase gene? Let us understand that. So suppose this is our vector DNA. Vector DNA. This is the gene which encodes for a protein which is known as beta galactosidase. This gene, this whole gene, it encodes for this protein beta galactosidase. But this gene also contains some restriction sites. Restriction sites. What are restriction sites? These are the sites where usually restriction enzymes cut the DNA and they help in inserting our desired gene in that plasmid DNA. So there are restriction sites present here and here. Now what happens is when we are inserting our gene of interest in this plasmid DNA, it will be inserted in between this gene. So this gene has been disrupted, the gene of beta galactosidase gene, because this whole gene from here to here, it encoded for beta galactosidase. But because this gene has now our desired gene as well, so the sequence for beta galactosidase, it has been disrupted. So this is known as insertion activation. Because of insertion of our gene of interest, this is our gene of interest. Because of insertion of gene of interest, this whole gene inactivated, and because of inactivation of this gene which encoded for beta galactosidase, there will be no beta galactosidase production in this host cell containing this plasmid. This is our plasmid. This plasmid will look something like this, the whole plasmid circular. So now what happens is, there is a compound which is known as X-gal. This X-gal, when it reacts with beta galactosidase, it gives a bluish color. This X-gal, it is usually colorless, but when it hydrolyzes our beta galactosidase protein, it gives bluish color. So all these colonies, all the bacterial colonies which have this plasmid DNA where our gene of interest has not been interested, only those plasmids will be able to produce beta galactosidase, right? So we'll know that wherever this bluish color appears in the colonies, we'll know that our gene of interest has not been interested in those colonies. And in the plasmids where our gene of interest has been inserted, only those plasmids, only those colonies will not be able to produce this beta galactosidase. Now what will happen? There will be no beta galactosidase for X-gal to react, so those colonies, they will not produce any bluish color. Hence those colonies will be colorless, or we can say we will have white colonies. So the question here says that those colonies which do not produce any color, which are colorless, those are the colonies which have our gene of interest, which have been recombinant, which have our recombinant DNA. What will be this plasmid called? The plasmid which have our desired gene is also known as recombinant DNA. And the cells which have our recombinant DNA, they are known as recombinant cells. So all of those colonies which are not going to show any color, with the type of cells which are going to be present there, they are going to be recombinant cells.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.